Vanakkam. Welcome to this video on biomedical instrumentation. In this series of videos, we've been looking at various types of sensors. In previous videos, we looked at resistive sensors. What are some of the resistive sensors that we looked at? We looked at thermistors, we looked at strain gauges, we looked at then we looked at capacitive sensors, then we looked at inductive sensors. Inductive sensors, we looked at LVDTs, for example. Then we looked at piezo sensors. In this video, we will look at electrochemical sensors. What is the purpose? The name provides some clues for us. Electrochemical sensors are those that convert chemical, or in this case, because we are interested in human health, they convert biochemical events directly into electrical signals or electrical events. That means they can be relatively fast, and because they are also electrical, they can be miniaturized to a large extent and highly accurate and highly sensitive and selective measurements of physiological parameters. In this case, physiological means physico-physiological, chemical, biological, biochemical. Essentially, these are used in so many critical diagnostic systems, such as blood analysis, blood gas analysis, studying the electrolyte concentrations and electrolyte compositions of blood or other fluids in a patient who is lying in the bed, or in other words, a bedside electrochemical monitoring, for example. Gas concentrations in breath, how much oxygen is present, the proportion of oxygen versus carbon dioxide, for example. Because if the person is not able to breathe, you will find a lot of their breath, their total gas found within the blood will become carbon dioxide, for example. An after effect of the inability to breathe, for example. So these are some of the, and maybe a necessity to track metabolites that are implanted inside the body. Following topics are of interest. Ion selective electrodes. We'll also be discussing about chemically selective FETs. FET is an electronic device like transistor. When we generally say transistor, we are looking at a bipolar junction transistor, BJT. Then there is a field effect transistor that is also called as FET. Those FETs that are chemically sensitive are called as ChemFETs. And those FETs that are used in the biological system are called as BioFETs, or biological field effect transistors. And then gas sensors that are used for electrochemical systems. Essentially, we are interested in measuring potential generated in ion selective or redox selective membranes. For example, in the case of ion selective electrodes, we are interested, for example, we can use this as an oxygen probe. as a potentiometric oxygen probe relative to some stable electrode, some electrode that is stable. The other approach is to use a method by which we measure a current by applying we apply a fixed bias, we apply a fixed potential, and then we measure a current as a function of the concentration of an analyte, for example, using some sort of reactions, Faraday reactions, such as oxidase reactions, biosensors, biosensors of, for example, glucose oxidase biosensing systems, Clark oxygen cells, for example. The other approach is to measure voltages or current to probe and measure redox kinetics or changes in conductivities. This is not quite often used, but the other two are used more frequently. A critical equation in these cases is the Nernst equation, which we will see in a future slide. The first topic of interest in this case is the ion selective electrode. The ion selective electrodes, the idea is to measure how much is the concentration of a particular ion in a given solution. When an ion selective electrode is immersed in a solution, usually a reaction happens between the ions in the solution and those on the sensor surface. So it's quite common. There is a reaction that happens between the solution ions and either the ions on the sensor surface or the metal atoms on the sensor surface. Usually, this leads to a situation in which an equilibrium is established. But when an equilibrium is established, there is a potential difference between the ions in the solution and the ions on the sensor or the electrode. And this is usually dependent on the activity of one particular species of or one particular set of electrons. So the output EMF is usually also influenced by other species. So these ion sensitive electrodes act as one half of the electrode, and the other half will be some reference electrode. In many cases, for example, we use a silver chloride electrode as a reference electrode. In some cases, we use a hydrogen electrode as a reference electrode. Most frequently, we are interested in measuring sodium and potassium, relative concentrations of sodium and potassium ions in blood serum. Very often, this is what we are interested in measuring. It turns out that an ion selective membrane produces or leads to a situation in which there is a boundary potential. There is a potential at the boundary between the two sides of a membrane that changes logarithmically with the change in the target ion. That is something that is what we discussed, for example, in using Nernst equation. I have discussed this in my other course, how we derive Nernst equation in my other course on neuromechanics, neuroscience of human movements. But this is also from basic chemistry and high school chemistry, for example. It's not really that tough to derive this equation, but for those who don't know how to derive this equation, you can check that. In a case where there is an activity of a single ion, which is very unusual in the biological case, there is almost always activity of multiple ions happening simultaneously in the biological system. But assume that there is only a single ion that is active, the monovalent ion that is active, then I could come up with a relationship between the potential at the surface, potential at the membrane, as a function of the relative concentration of the ion, for example. And this obviously is dependent on gas constant, temperature, and Faraday's constant R, T, and F. And at a given temperature, say for example at 25 degrees Celsius, I could substitute for other values such as R and F, and I could come up with values of uh, the rest. So I could essentially compute the concentration of a given species of ion. I could compute the concentration of a given species of ion using this equation, for example. And there are different types of these ion selective electrodes that we see, and some of these are useful for certain to measure certain types of certain ions. Some of them are used to measure chloride ions, some of them are used to measure potassium, etc. And again, once you use a reference electrode such as the silver electrode as the reference electrode, then I can make the measurements with millivolt accuracy, except that I will still need a high impedance processing circuitry. Some solid state materials, for example zirconia, are very sensitive to oxygen, and they can be used to provide and measure electrochemical response when they react with oxygen. So this way we can actually use it to measure concentration of oxygen. So that way zirconia at high temperatures, zirconia becomes an oxygen ion conductor. So when it is exposed to gases that contain oxygen, then the oxygen molecule will leave and form the O2 ion. These ions will move through zirconia to another electrode, thus releasing electrons. And this movement will usually generate a voltage that is described by this form of Nernst equation, for example. The other topic of interest is the ChemFETs, or the chemically sensitive field effect transistors. In this case, what happens is that we use field effect transistors. There is a gate that is present in the field effect transistors. If we modify the gate of a field effect transistor with a ion selective membrane, for example, you essentially end up with what is called as a chemically sensitive field effect transistor. Note that field effective transistors are excellent amplifier with high stability from the electronics viewpoint. So charged species at the membrane of the surface, which is acting as the gate, or the gate channel will generate an electric field, will change the threshold of the gating mechanism such that the drain current to the concentration, the transfer function, therefore we can that transfer function will then be logarithmic per specific ionic species. Thus it will essentially represent an electronic mechanism of the electronic counterpart of the Nernst equation, for example. That way I can actually straight away measure the concentration transfer function, concentration of a given ionic species, right? So the gate of the FET is replaced with the ion sensitive layer, and is used in the solution. So when we do that, the target ions will interact with the gate, and the change in the surface potential changes the channel conductivity. So note that they have high input impedance and are highly responsive. So, and because these are also FETs, we can miniaturize this at a high level, and they are suitable for lab on chip and for biomedical devices. Lab on chip means the entire device is in a single chip. That's about ChemFit. The other topic is the BioFit. This essentially couples a semiconductor channel modern topic. That is, it uses graphene or a silicon nanowire or CNT, carbon nanotube, that is used as a layer to recognize biological species such as antibodies, aptamers, enzymes, for example. In these cases, these will bind to target biomolecules and modulate the surface charge, thus changing the channel conductance in relatively high speed, or with near real time effect you could find. So these are some of the other applications. For example, point of care diagnostics quite frequently use biofit swabs. For example, for COVID-19, you could use that. They are also used to measure metabolite sensing and organ on chip, for example. And then we can also use similar principles to analyze gas concentrations. For example, Clark oxygen cell, a galvanic fuel cell, potentiometric zirconia for these, and what are some of the performance factors in these cases? Cross sensitivity, what is going to be the lifetime of these sensors, or this. With this, we will end this topic, and we will continue our discussion on optical sensors in the next video. Thank you for your attention.